Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, how are you all? I hope you all are doing well and enjoying your studies with Pakistan International School Thai's virtual learning system. Dear students, today's class of biology is for grade first year, and in that we are studying chapter number 6, prokaryotes. We have already started chapter number 6, in which we will talk about prokaryotes. As you know, prokaryotes are the organisms which don't have the proper nucleus, which is enclosed by a nuclear membrane. We have started prokaryotes, in that we have studied before archaea domain, and now we are studying bacteria. These two are prokaryotes organisms, and they are the primitive, or we can say the ancestors of eukaryotes. Now we will see today about the structure, in which we'll talk about shape and size of bacteria and its complete structure. Its model structure we have studied in chapter number 1, but right now we will see some very important features other than its internal structure. So let's get started. This is a typical bacteria, in which you can see all its parts which are present in its structure. Today we will talk about its external structure, specifically cell wall, because up to cell membrane all bacteria are same. Sometimes cell membrane is also included in its if we talk about this structure, so the cell membrane is included in the internal structure, not in the cell envelope. But specifically today we will talk about its cell envelope, in which we'll talk about a cell wall and its outer covering, that is called glycocalyx. And then we'll see its size and shapes and motility, etc. So this cell membrane is actually its outer layer, but we have discussed this in chapter number 1. So right now we are talking about glycocalyx. Glycocalyx is actually the external structure of polysaccharides or polypeptides outside the membrane. As we know that cell wall of the bacteria is made up of polypeptide or polysaccharide, or we can see some other structures we will talk about that is not made up of cellulose. So glycocalyx is the outer covering or outer structure which is made up of polysaccharide, very outer than cell wall. We are not talking about this cell wall, we are talking about its external covering other than cell wall, because lots of bacteria are having this type of structure that is called glycocalyx. This glycocalyx can exist in two forms, like in the capsule form and in slime layer form. Glycocalyx layer that helps to protect bacterium and keep it from being phagocytosed, that is considered as virulent factor. If we see here in this structure, that is a capsule. So capsule is actually, it is a condensed layer that is tightly associated with underlying cell wall. Tightly associated. So that is attached with a cell wall. Tightly attached with the cell wall, and gives the sticky and gummy appearance to the cell. So what is the benefit of this one. Like it makes the sticky appearance on the cells of the body. Like for example, T cells or macrophages, which phagocytose uh, these type of cells for immunity purpose. So that is very helpful for the bacteria over there, but they are not phagocytosed properly because this capsule is having the sticky appearance, so it just stick together. So lots of cells of bacteria stick together and they form biofilm like structure, like a layer. They just found as like all the cells like this, they stick together and they form the layer on any surface. So that is hard to phagocytose or by macrophages. So that is considered as virulent factor, like it can cause more diseases, and it can be antibiotic resistance as well. Then we can see the next one, that is called slime layer, that is thinner and less tightly bound to the cell wall, that is loosely bound. So it prevents the cell from drying out, especially you can see here the slimy appearance of this slime layer. That can also be said to capsule, but that is actually the slime layer. So that traps nutrients near the cell also, that helps to stick the cells together also, and that adhere to object. They can help bacteria adhere to objects in the environment. So there are some common examples in our body also, like plaque, which is formed on our teeth. That is actually the slime layer of Streptococcus mutans, and a few anaerobes, fusobacterium and actinobacteria. They do the process of plaque, making the plaque and destroy our teeth. So many prokaryotes secrete it on their surfaces. What glycocalyx is secreted on their surfaces? It's meaning sugar coat. So that's why that is made up of polypeptides or polysaccharides. Next, move on to the bacterial cell wall. Bacterial cell wall, that is, we can say, specific in different type of bacteria. So you can here see the structure of capsule. Under that, this is all bacterial cell wall. It is also having like flagellum and pili, and we can see some of like different layers in this cell wall. So let's talk about this. Bacterial cell wall actually is made up of peptidoglycan. So molecule found only in bacterial cell wall. We know that in plant cell wall, that is made up of cellulose. Chitin is the major constituent of fungi cell wall. So peptidoglycan is the constituent of a bacterial cell wall. That is the specificity of bacteria. The rigid structure of peptidoglycan gives the bacteria its cell shape, surrounds the plasma membrane, provides prokaryotes with the protection also. So from bacterial cell wall, there is cell membrane. From that to the inside, all the cell is the same. If we talk about specifically about the cell wall, so there is the different categories of cell wall. If we just classify or divide the bacteria, so they are divided on the category of cell wall. Specifically, they act as gram positive and gram negative. There are two types of structures, two type of bacteria are present in the um like in bacteria category. So let's talk about both of them one by one. Gram negative. Negative means does not. So what does it mean? Like gram negative. First we'll talk about its structure, then we'll say what is gram negative and gram positive. Why it is said to be gram negative and gram positive. If you see this bacteria, if you take its cell wall, so you can see it is having different types of layers. The innermost is having the cell membrane. It is lipid bilayer, we know that. Then it is having specifically this layer. This is called peptidoglycan layer. This layer is having specificity and means it is different in different bacteria. Then on that we are having outer membrane layer, and that outer membrane layer is having further some other substances present over there, lipopolysaccharides, then foreign proteins, phospholipids, as well as there is the space between these two layers. So if we see, first one is lipoprotein. This lipoprotein layer connects the peptidoglycan to the outer membrane. This lipoprotein disconnects the peptidoglycan, this layer, with the outer layer. Outer membrane proteins target sites for antibiotics. Outer membrane, these are for antibiotics. They target this one because this is the connection. The by that connection is disturbed, and this cell wall is degenerated. Lipopolysaccharide, that layer, this consists of lipid A, like lipoproteins, as well as attached polysaccharides over there. You can see that's why it is called lipopolysaccharides, because it is attached with lipoprotein and polysaccharides are there. Between these two layers, there is the space that is called periplasmic space, as well as between these inner membrane and the outer membrane. This is inner membrane, that is called cell membrane. The outer membrane in between this is the periplasmic space. Peptidoglycan, thin of peptidoglycan, this layer is thin of few layers, 7 to 8 nanometers just. So it contains the dry weight of about 10% or less than this of gram negative strains. In gram negative, you can see here lots of proteins, lots of like different substances which are present in cell wall, gram negative cell wall. First, phospholipid, that is the first layer, cell membrane. Then there is peptidoglycan layer, that purplish one. You can see here the periplasmic space, and here also the periplasmic space, like that is the plasma simple fluid is there. Then this is lipoproteins, which are connection with the peptidoglycan and the outer membrane. There are certain lipopolysaccharides also are embedded in that, you can see here. And some proteins are there which are embedded in this one. Uh, but there is very specific thing that is called porins. Porins are the holes that are made up of proteins also. So these porins, they act like pores for particular molecules to come inside this layer, inside the bacteria. Gram-positive bacteria, if we see, that possess thick cell wall containing many layers of peptidoglycan. You can see here. This is inner layer, phospholipid layer. These are the carrier proteins. Then you can see 1, 2, 3, 4, and 5. Many layers of peptidoglycan. And very, very important, that is called teichoic acid. Teichoic acid or lipoteichoic acid. This is lipoteichoic acid, which is having the connection with a joint with the phospholipids, and there is another teichoic acid just that is called that is connected with the peptidoglycan layer. Gram-positive bacteria, they are having peptidoglycan is the outermost structure and makes almost 90% of the thick compact cell wall. What is the difference between gram-positive and gram-negative? That gram-positive bacteria, they are having a thick polypeptide layer because it is having lots of layers. So gram-negative, they are having just a few nanometer that is thick. Teichoic acid is present in gram-positive, that makes cell wall more acidic, but that is absent in gram-negative. Lipopolysaccharide, that is absent in gram-positive, but that is present in gram-negative. Outer membrane proteins, and those are called the porins. They are absent in gram-positive, but they are present in gram-negative. Now we will see that why we say that to gram-positive and negative due to the gram staining. The staining process of different types of bacteria just lead to this technique by which we can distinguish between different varieties of bacteria, which is virulent and which is less virulent and which is non-virulent bacteria. So that technique was developed in 1884 by Danish scientist Christian Graham. So that scientist, that after the name of this Graham scientist, that is said to be Graham staining. It is differential stain, like we can differentiate between bacteria. And first of all, its first step is to stain and the bacteria, the film, or we can say the culture of bacteria, with crystal violet color. Crystal violet color, you can see here, that are stained with crystal violet color, then treated with a mordant or solution fixer that fixes the stain inside the cell, that is iodine. Now you can see crystal violet color when it is all bacteria stained. So these bacteria are just retaining the color purple or violet, but later on it is treated with mordant, which fixes this color, so all purple. Here is we can say the midpoint, and the very important, when we treat that culture with the decoloring agent, definitely that is alcohol. So after treating alcohol, we can see that it decolorizes, or we have to wash that bacteria with alcohol to remove all the colors. Later on in the last, that is counter-stained with safranin. Safranin is a light red dye. So all the bacteria are just treated with safranin. So what is the result? After treating safranin, we say that some bacteria which do not retain the color, when we have treated that with alcohol, so that they get decolorized. So when they get decolorized, so when we treat it with safranin in the later stage, the last stage, so they get a red color on that. So some bacteria didn't get saffron red dye, but some bacteria got this color. So because before with treating alcohol, they got colorless, so that's why they again stained with a saffronin and they retain the saffronin color. Now, this is actually the result in which they find that he found that at the time that the bacteria which did not get saffronin color, so they are called gram positive because they got purple color and they did not leave the color while decoloration with alcohol. But the red one is gram negative because they have decolorized with alcohol and then they got saffronin stain on that. So what does it mean that gram-positive bacteria stained dark blue or violet by gram staining, and they are not getting the retained color of that is the important one, the crystal violet. We have to just uh, stain with the bacteria with crystal violet. If they got retained the color crystal violet, they are positive. If they are not, they are negative. So that is called gram positive and gram negative bacteria. Why it is happened? Because gram positive bacteria have more peptidoglycans than do gram negative bacteria, so they retain the original violet dye and cannot be counter stained. So if you see, if you see in this gram positive structure of bacteria, there are lots of peptidoglycan layers over there. So alcohol cannot get deep inside to just decolorize it. So it retains the purple color. It does not remove all the color, so it retains the violet stain. But in gram negative, that it is having just one layer of peptidoglycans, or the colors are decolorized and they retain the saffronin color. So that's why they are said to be gram negative bacteria. Gram positive bacteria are actually more virulent because they are having gram positive is more virulent because they are having lots of peptidoglycan layers over that, which makes the cell wall more stronger, and it is harder to break that, or it is harder to dissolve with any of the antibiotics. So they are more antibiotic resistant. Now we will see size of bacteria. So they are smallest living creatures. So most bacteria minus 0.0 micrometer to that is 2 to 8 micrometer in length, and but 1/10 the size of eukaryote cells are typically 0.5 to 5 micrometer. So that is more than this one. So giant bacteria are also there. Thio, Margarita, Namibiensis, and all Titanospirillum namibiensis, like this species, and Epulopicium fischelsoni. They are also almost half an mm long and are visible to the unaided eye. They are giant enough. So there are some smallest bacteria are member of the genus Mycoplasma, that is zero point 0.2 to 0.25 micrometer. That is too small size, like that is the smallest size we can say of the bacteria. So E. coli, that is having almost 2 micrometer long and 0.5 micrometer in diameter, and Staphylococcus aureus, that is the rounder spherical one, 1 micrometer in diameter, rod shape, Bordetella pertussis, that is causative agent of whooping cough, so that is having this type of diameter and in 0.5 to 1 micrometer in length. Corkscrew, that is Treponema pallidum, causative agent of syphilis, that is 0.15 micrometer diameter and 10 to 13 micrometer in length. So if we just see some that is relatively large, like Azobacter, which has diameter 2 to 5 micrometer, that is too much, and Cyanobacteria, they are having 6 micrometer to 12 micrometer. Acromatium, that is having 5 micrometer to maximum length of 100 micrometer depending on the species, that is too much, like the size that can be easily seen through the microscope, electron microscope, as well as highly defined light microscopes. Now we will see shape of the bacteria. They are found in varieties of the shapes, like some are spherical, that is called coccus or coccus. Some are bacillus, they are round from the ended and cylindrical shaped. Vibrio, they are having the comma shaped like this, and they do vibratory motility. Some are spirilla, like they are having coil or spiral shaped. So spiral like this, sorry. And spirochetes, they are long, cylinder, flexible, but they are having spira means coil, so they are coil shaped, and chaetae mean hair. So they are, because they are very thin, so the coiled hair, curly hairs. So filamentous, they resemble radiating to the sunlight. So they are just filamentous, the long one. So Coccobacilli, in which the two categories, Coccus and Bacillus, both are just shown in the same category. Like they are short enough, they are so that don't make Coccus and don't make Bacillus structure. So they collectively called Coccobacillus. Then tetrahedral cuboidal shapes are also be found. Tetrahedral, in which the four sides are there, and they also appear in the cuboidal shapes. They are star shaped also, rod-like shaped also, and they are like they can live anywhere. Yeah, we know about that. So we will see cocci, that is having different type of structures, like diplococci, two, or when just two cocci, just they are present together, they joined together. Then streptococcus, they make a rod like structure by like beaded like structure. Then tetrad, they arrange in 4 cells when the art, that is sarcina or sarcinae, they formed like tetrad, when two tetrads just joined together, and you can see here they form crystal-like structures. And staphylococcus, like all the lots of cells just form the ball-like structure and found in clusters. In bacillus, you can see single bacillus, diplobacillus, like two bacilli are just joined together. Then streptobacilli, they are joined together in long filaments like structure. You can see here the original high definition structure. And coccobacillus, like they are not bacillus and not coccus, but a smaller than bacillus form, rounded from both of the side. So there is another structure that is called a spirae, that is Vibrio. You can see Vibrio that is having this like common like structure, then spirillum. You can see spiral rotatory, and then like this spirochetes structure. Spores and cysts, that is very, very important term regarding this bacterial structure. They are found outside of the cell wall and inside also. For we can see to bear unfavorable conditions. So by making this, they can live for thousands and thousands of years in unfavorable conditions. So they are like the outside coverings they can make in the form of spores and cysts. So some bacteria survive unfavorable conditions, so dormant forms are metabolically inactive and germinate under suitable conditions. So the first one is endospore, that is dormant, a tough and non-productive structure of small bacteria. Small number of bacteria, they can do this one, and the primary function is to pass through the unfavorable conditions. They can be well resistant to ultraviolet, gamma radiation, desiccation, lysozyme, temperature, starvation, chemical disinfectants also, and they can be found anywhere for a longer period of time, like Clostridium tetanus virus and gangrene and bacillus also, which produce anthrax. Cyst is also found in the dormant condition, but unlike endospores, they are resistant to heat at high temperature, but they are not. Endospores, they do like appear in this high temperatures. So you can see the formation of endospore here. First of all, this is the vegetative cell which can reproduce. Then this DNA is duplicated. So the copy of DNA just get at a side, and a tough substance of cytoplasm is just going to start to move around this DNA. Actually, the reproduction is just on in the DNA, so DNA has going to be protected. So other DNA, the old one, as well as all the cytoplasm gets degenerated, like dehydrated as well, and this spore can be released from the cells, and it can be it can live for thousands of years like that, and in favorable condition, then it again can be grow into vegetative cell. So exospore is formation of the filament that is not present in that is not formed in the cytoplasm, but out of the cell wall, that makes certain cells like this. This is the cytoplasm gathering in separate places. So in that, the DNA is also present, and they just form the cluster or the filament like a structure. And then after in favorable condition, they rupture and produce lots of cells. So that is actually exospore is for the reproduction purpose as well. So there is the cyst formation. In cyst formation, we can see here outside the cell wall, there is a cyst of Azobacter. If you just see the cyst deposition of layers and layers of cell wall around the cell, rather within the cell in case of spores. In the spore, cell wall is just gathers around the DNA inside the cytoplasm in endospore formation, but here in the cyst formation, cell wall just gathers out of the cell wall, lots of layers, and cytoplasm gets we can say that gets decreased. And myxobacteria and azobacteria, they are they do this process in which they involve in the nitrogen fixation, and they give protection also. So we have seen before the endospore formation and exospore formation. So budding at one end of the cell, that gets make the filament, and exospores do not contain dipicolinic acid, and they get resistance against the desiccation and heat, but not the higher temperature. Now we will see some types of motility in bacteria that depends first of all flagella. Flagella is a very important part of that bacterial cell wall that like for the movement of bacteria from one place to another. So when they are also classified on the presence of flagella. If one flagella at the one side, so that is called polar or monotrichous bacteria. If lots of flagella are present at one side, they are called lophotrichous bacteria, and at unipolar also. Bipolar but monotrichous, we can say the one, not monotrichous, actually one flagella on both of the sides, that is called amphitrichous. When in peritrichous, flagella are all over the body, and in amphilotrichous, that is tuft of flagella at both of the ends are present. Amphilophotrichous. You can see here the original microscopic diagrams of that, how they are present. Now we will see structure of flagella in the movement and motility, how they move, because that is having a little complicated structure. That is, flagella consists of a basal body. It consists of three parts. Basal body, hook and main filament. So this is basal body, which is a basal body and or granule that is 200 to 500 in the diameter angstrom, we can say it. And in gram negative bacteria, basal ring has two sets. You can see here the structure of gram negative bacteria, but in gram positive bacteria it is having just one ring. So in these two sets are having proximal and distal end. So two rings are completely four rings. We can say two in each set. That is M ring, S ring, P ring, or L ring. So that is a detailed view of that one, but you have to just remember the concept, like how they move and how they are adjusted in the cytoplasm. So the basal body is adjusted in the cytoplasm. So the one ring of that is in the cytoplasm, and it consists of two pairs of discs in gram positive and four pairs of discs in gram negative, and in gram positive only one pair of discs. Like this is two pairs of discs, one in just gram positive bacteria. So they are connected with a central rod. This is the central rod. You can see the central rod. These are the inner rings. These are M ring and S ring, and they are the basal body anchors the flagellum into the cell envelope. We can see that is keep that inside the cell. That is, we can say, the root of the flagellum. The hook is curved portion. You can see here the hook. So this other pair of outer rings, that is PLN ring, they are adjusted in the other membrane, outer membrane of the cell wall, as well as peptidoglycan, because that is gram negative bacteria. Then this hook is the curved part, and it connects the basal body and originates and the filament. It is projected from the cell surface. Like this is cell surface, it projects from outside. Then the filament is a hollow structure which originates from the hook, and the filament is composed of globular protein and flagellin. That flagellin just twisted around, like we have seen the structure of this DNA, like it is twisted around like this to form the helical structure, but deep inside the core is empty. Several intertwined chains of flagellin that form a helix around a hollow core. So how does it move? How does it move? We can see like when this basal body rotates around this area, so then it like rotates this rod, and then hook rotates like this, and flagella rotate. That moves by rotation, and that bacteria moves forward. This is gram positive bacteria, this is positive, and this is gram negative bacteria. Now we will see its other movement, that is called spirochetal movements. In spirochetal movement, that spirochetes, how do they move? They are also having endoflagella, that is called axial filament, but they are present inside the outer membrane, beneath the membrane. So that definitely they are present in gram-negative bacteria. They are present inside, they are not appear outside the body, outside the membrane. So their basal body are adjusted like before we have seen in the cytoplasm, and then they twisted around this membrane like this, and over there there is outer membrane. So you can see lots of endoflagella or axial filaments present there. This is in this periplasmic space. So then they move. They can perform a flexing, swimming, creeping, and spinning type of movement with the help of this axial filament. Now you can see here in this electronic microscope diagram the gliding movement of the bacteria. That is another type of motion or motility of bacteria. So they move on the solid surface, and the cells of bacteria, they produce a slime. So slime trails, like a snail is moving on the solid surfaces, and first it releases or secretes slime, then it moves on. So like that, they also do the same process. They release slime trails, and then cells glide in groups singly, or they can reverse also. So you can see here they can reverse also. Example is myxobacteria, flavobacteria, and cyanobacteria are having this type of motility. You can see here the slime trail secreted by bacterial cell wall. So then they glide over it and then move forward. Dear students, that's it for today. I hope that is like you found it interesting and you understood all the like characteristics of bacteria. Inshallah, in the next lecture, we will continue the topic bacteria. Wish you all the best. Allah Hafiz.